Laboratory of Genome Informatik. Moderator pada hari ini yaitu Bapak Wayan Aditya Swardiana M.Kom, peneliti Pusat Riset Komputasi BRIN, para periset di lingkungan BRIN, dan peserta kolokium yang berbahagia. Alhamdulillah, puji syukur kita panjatkan karena berkat rahmat Allah Subhanahu wa Ta'ala, Tuhan Yang Maha Esa, kita dapat berkumpul secara virtual untuk menghadiri acara kolokium Mahameru Lecture Series pertama dengan tema Pengenalan Mahameru HPC, Laboratorium Komputasi Kinerja Tinggi BRIN pada hari ini Kamis 13 Juli 2023. Baik, pengawali acara pertama, kita akan mendengarkan secara hikmat lagu Kebangsaan Indonesia Raya. Kami mohon kepada Bapak Ibu untuk mengambil sikap sempurna. Kami persilakan kepada panitia untuk menampilkan lagu Indonesia Raya. Baik Bapak Ibu, berikutnya kita akan foto bersama. Kami mohon kesediaan Bapak Ibu untuk menyalakan kamera dan memberikan senyum terbaiknya. Kita akan mulai sesi foto sesuai dengan aba-aba saya. Silakan untuk membuka kameranya Bapak Ibu. Dan panitia akan melakukan tangkap layar. Baik, saya mulai dengan hitungan 1, 2, 3 untuk layar pertama. Dan layar selanjutnya akan dilakukan tangkap layar oleh panitia. Baik, terima kasih Bapak Ibu. Selanjutnya memasuki acara inti, yaitu sesi pemaparan dan diskusi yang akan dipandu secara langsung oleh moderator pada hari ini, yaitu Bapak Wayan Aditya Swardiana M.Kom, peneliti Pusat Riset Komputasi BRIN. Waktu dan layar kami silakan.
Baik, terima kasih Bu Yetty. Perkenalkan, saya Aditya dari Pusat Riset Komputasi. Hari ini kita akan melakukan Mahameru Lecture Series pertama dengan topik pengenalan Mahameru HPC BRIN. Untuk narasumber pertama dari Dr. Rifki Sadikin M.Kom sebagai Kepala Pusat Riset Komputasi akan memberikan pengenalan Mahameru HPC BRIN, ELSA HPC BRIN, pengelolaan sumber daya komputasi dan komite penggunaan HPC BRIN. Waktu dan tempat saya persilakan kepada Pak Rifki.
Terima kasih Bu Yetty dan Mas Adit atas pembukaan dan tadi perkenalannya. Yang terhormat Bapak Ibu semua, partisipan di acara ini. Hari ini kami akan memperkenalkan infrastruktur riset di BRIN yang merupakan infrastruktur komputasi ya. Jadi saya akan membawakan dalam bahasa Indonesia. Jadi I'm sorry to Ajay, I will deliver in Indonesia and you can deliver in English later on. Untuk sesi kali ini saya akan memperkenalkan infrastruktur baru yang ada di HPC BRIN dan juga konsolidasi komputasi ya. Nanti mungkin yang yang beberapa sumber daya komputasi yang ada di BRIN itu sedang kami lakukan integrasi dan konsolidasi. Kemudian nanti bagaimana pengelolaannya dan juga penggunaannya. Jadi saya akan share screen-nya. Sebelum itu, perkenalkan nama saya Rifki Sadikin, saat ini Kepala Pusat Riset Komputasi, dan juga Ketua Pelaksana HPC BRIN. Nanti saya akan jelaskan struktur pengelolanya, karena saat ini sifatnya cross unit ya di BRIN, jadi tidak cuma satu unit saja yang mengelola, tapi juga beberapa dari kedeputian yang berbeda dan pusat riset yang berbeda, juga pusat yang berbeda. Jadi agendanya pertama, saya akan memperkenalkan sumber daya komputasi BRIN yang saat ini, termasuk yang diadakan pada tahun 2022, dan kemudian di-deploy tahun 2023, dan akan kami buka untuk publik setelah nanti komite pengguna ataupun setelah selesai dalam persiapan kurang lebih minggu depan ya, ataupun 2 minggu lagi kita akan buka melalui ELSA. Yang kedua terkait dengan sistem pengelolaan dan penggunaan, ini yang mungkin SOP umumnya akan saya jelaskan, penggunaan HPC BRIN. Dan yang ketiga, rencana diseminasi ataupun community building ya, karena ini yang paling penting, karena kami ingin agar pengguna itu makin expert dan kita sama-sama bisa mengeksploitasi secara positif ya sumber daya komputasi ini untuk kebutuhan riset. Jadi kalau HPC BRIN itu merupakan infrastruktur riset yang saat ini merupakan hal yang menjadi enabler untuk beberapa riset yang mendasar ya. Jadi kalau kita baca-baca terkait infrastruktur HPC di dunia, tentu saja sekarang saat ini mengarahnya kepada skala eksa. Nah, tapi tentu saja kita di BRIN tidak mungkin, atau di Indonesia ya, tidak mungkin mulai langsung besar, karena kita butuh juga menumbuhkan komunitas, sembari nanti kalau komunitasnya makin besar, sumber dayanya juga kita tambah. Tentu saja skala ekstrim HPC ini akan membuat beberapa riset itu menjadi mungkin dikerjakan. Misalnya nomor 1 terkait dengan bioinformatika, bahwa saat ini pendekatan in silico, ya, misalnya molecular modeling dan simulation itu menjadi hal yang lumrah, dan ada fase-fase yang membutuhkan high performance computing, misalnya molecular dynamics gitu. Jadi beberapa kasus lain misalnya di material science itu juga menjadi hal yang lumrah menggunakan sumber daya komputasi untuk pemodelan sebagai salah satu cara penemuan material baru. Selain itu ada beberapa kasus riset lain, ya, misalnya terkait dengan engineering, terkait dengan pemodelan iklim dan cuaca earth science ya, ataupun oceanografi, itu juga saat ini membutuhkan sumber daya komputasi yang besar. Misalnya kita akan melakukan simulasi cuaca, melakukan simulasi iklim, urgent computing, misalnya kalau terjadi kebencanaan, digital twin-nya bagaimana supaya bisa cepat responnya dan sebagainya. Yang terakhir itu adalah terkait dengan riset-riset yang berbasis machine learning ya, yang untuk kecerdasan artifisial itu juga membutuhkan infrastruktur komputasi yang besar. Saat ini infrastruktur komputasi seperti GPU base itu banyak digunakan untuk pendekatan data science ataupun machine learning. Nah, di sini Mahameru BRIN berusaha untuk bisa setidaknya menyediakan layanan ataupun sumber daya komputasi untuk riset-riset yang ada di Indonesia, supaya menjadi bisa melakukan riset-riset yang membutuhkan komputasi besar. Jadi ini lingkupnya banyak ya, dan karena itu kami nanti berencana dengan arahan Kepala BRIN kemarin membentuk komite pengguna agar bisa bersama-sama menumbuhkan komunitas dan juga menentukan kelayakan sebuah kegiatan riset untuk penggunaan infrastruktur komputasi ini. Jadi kurang lebih seperti itu. Kemudian yang ada di BRIN ini seperti apa? Tentu saja kita sama-sama tahu ya bahwa BRIN merupakan lembaga dari integrasi dari beberapa lembaga riset yang dahulu, dan pada umumnya waktu dahulu kita punya klaster-klaster yang kita sebut klaster HPC itu di beberapa tempat. Jadi ada yang di Serpong, ada yang di Bandung, ada yang di Pekayon, dan bahkan ada yang di Yogyakarta. Nah, jadi bagi pengelola itu akan kesulitan kalau banyak tempat. Selain itu tidak efisien ya, listriknya di semua tempat, pendukungnya di semua tempat, dan seterusnya. Jadi kami mengusulkan waktu itu sumber daya itu dikonsolidasikan agar efisien ya, listriknya di satu tempat, pendukungnya, karena kan kalau cluster server itu harus ada pengatur suhu dan kelembaban, kemudian ada listrik yang di backup dengan UPS dan juga genset. Nah, ini akan lebih mudah kalau dikonsolidasikan. Dan kita juga bercermin di best practices di negara-negara lain yang memang melakukan konsolidasi sumber daya komputasi. Tentu saja kalau kita lihat dari kapasitas, saat ini kita di BRIN belum masuk ya ke 100 besar di HPC dunia. Tentu saja kita, tapi kita perlu ada titik mulai agar kita bisa punya infrastruktur komputasi yang mumpuni. Nah, skematiknya adalah kita berharap nanti data center itu, ini data center tanda kutip, infrastruktur HPC itu memiliki 2 pusat saja, yang satu di Serpong dan yang satu lagi di Cibinong. Yang di Serpong itu kita akan buat sebagai disaster recovery center, begitu juga kebalikannya ya, sehingga nanti kalau ada apa-apa data itu terbackup di satu sama lain. Dan ini kami sedang proses juga bersama-sama Pusdatin BRIN. Sehingga nanti sumber daya komputasi itu dikonsolidasikan di satu tempat. Saat ini sudah berlangsung ya, dan beberapa sudah dipindahkan di Mahameru, di Gedung Lab Indrajauh. Jadi lokasinya di Gedung Lab Indrajauh KST Soekarno di lantai bawah. Nah, ini yang HPC 2022 ya. Jadi saya perkenalkan HPC 2022 itu pengadaan tahun kemarin. Itu ringkasnya ada 92 computing nodes, masing-masing itu dual dual prosesor dengan RAM 256 GB. Kemudian punya satu high memory node ya, yang memorinya sampai 1,5 terabyte. Kemudian ada 2 login dan management node, dan juga ini saya belum tambahkan storage, dan storage yang totalnya 4,5 petabyte bisa terpakai. Semuanya terhubung di InfiniBand, jadi high performance connection. Jadi ini HPC 2022 yang sampai bulan Juni atau Juli itu masih deployment ya, dan makanya kita belum launching. Tapi saat ini sudah cukup siap, nah masih di trial oleh teman-teman admin, sehingga nanti seminggu 2 minggu ke depan kita bisa buka di Elsa. Nah, kenapa di sini tidak ada GPU gitu ya? Kita waktu itu sudah usulkan sebenarnya ada GPU cluster, tapi arahan dari Kepala BRIN bahwa GPU cluster yang kita punya itu dimaksimalkan terlebih dahulu penggunaannya sebelum kita lakukan penambahan. Pada dasarnya kita diberikan infrastruktur ini berharap nanti banyak penggunaannya, jadi okupansinya tinggi, sehingga kalaupun ada permintaan kita punya basis bahwa penggunaannya sudah penuh. Jadi ini yang 2022 ya, yang vendornya itu Lenovo. Sebenarnya kita punya banyak ya, ini yang saya ceritakan tadi, kita konsolidasi klaster-klaster yang ada di BRIN yang tersebar di beberapa tempat, dan ini mungkin saya akan ceritakan yang GPU cluster ya. Yang GPU cluster itu kemarin ada di Serpong, tapi sudah dipindahkan ke Cibinong, yaitu DGX A100, DGX 1, dan IBM Watson. Ini kalau kita lihat detailnya masing-masing DGX A100 ini punya 8 card A100, bisa dipartisi jadi kurang lebih berapa ya? 70, saya lupa. Nah, 70 atau berapa partisi gitu. Dan ini sebenarnya sudah punya 2 computing nodes dan punya storage 300 terabyte. Lebih kurang dari 300 terabyte ya, kurang lebih 293. Dan saat ini sedang fase perbaikan ya, karena terakhir ada kejadian kerusakan dan kami sudah hubungi vendor, saat ini dalam proses perbaikan. Nanti kalau sudah diperbaiki, kita buka juga infrastruktur ini. Kemudian DGX 1 yang isinya 8 card GPU V100, ini NVIDIA punya, ini juga sudah kami bawa, sudah dipindahkan ke klaster di Cibinong, supaya nanti bisa mengisi di sini nih. Jadi akan jadi satu sistem sehingga punya akses ke storage dan juga punya akses ke computing node. Kalau yang kemarin itu masih island sendiri di DGX 1. Dan yang terakhir yang IBM Watson ini juga dalam proses revitalisasi agar bisa digunakan oleh teman-teman yang punya kebutuhan riset kecerdasan artifisial gitu ya. Jadi kita berusaha agar beberapa infrastruktur ini tidak pemanfaatannya optimal ya, kita tingkatkan. Kemudian ada beberapa server lain yang berasal dari XLP, yaitu Dell Big Data Nodes yang storage-nya sampai 2,8 petabyte. Kemudian ada HP ya, 24 computing nodes. Ada juga GPU card 10 yang P100 dan memiliki juga GPU untuk render. Nah, ini juga akan dipindahkan ke lab indera jauh, karena yang kalau ada punya dua kita jadi repot ya. Ini dalam proses pemindahan. Jadi yang saat ini ada di ELSA itu menggunakan yang klaster ini ya, yang HP 24 computing node, karena itu selalu penuh. Jadi ini sedikit apa ya namanya, sumber daya komputasinya terbatas. Nah ini rencananya sedang kami pindahkan, lagi sedang di backup semua datanya, sehingga nanti kalau ada pemindahan itu data tidak hilang. Beberapa server lain itu juga rencananya akan dibuka juga dan direvitalisasi ya kalau misalnya belum ada penggunaan. Misalnya ini cluster blade server yang sebenarnya kalau tidak salah pengadanya 2020-an ya. Dahulu ada di Eijkman. Ini juga mau kita satukan dan nanti terbuka untuk digunakan. Kemudian ada rak junjunan. Ya kalau teman-teman X8 mungkin mengerti di sini dari junjunan sudah kita pindahkan, juga dari Pekayon. Nah ini maksudnya proses ini terus berjalan integrasi supaya sumber daya komputasi ini bisa kita manfaatkan bersama. Sedangkan kalau infrastruktur jaringannya ya, infrastruktur jaringannya itu dedicated 100 Mbps keluar, baik dari internet maupun internal BRIN, sehingga cukup cepat ya, nanti tidak perlu khawatir kalau misalnya kebutuhan transfer data yang cukup besar. Nah ini atas bantuan dari Pusdatin BRIN, kita punya alokasi koneksi yang dedicated untuk HPC. Ini beberapa skematik yang apa ya? Ya ini belum sedang dalam proses ya. Jadi selain HPC nanti digunakan untuk kebutuhan riset, kami juga buat agar mendukung layanan laboratorium, terutama untuk infrastruktur penghasil data besar. Jadi misalnya di sini di Gedung Genomik dan Cryo-M itu ada sequencing dan Cryo-M yang menghasilkan data besar. Dan ini sudah ada sambungan 10 gig dari lab ke HPC. Namun flow-nya belum diimplementasikan ya. Jadi nanti salah satu yang kami akan dalam proses ya untuk menyediakan flow sampai penyimpanan di storage yang ada di HPC. Sedangkan kalau untuk integrasi node-node yang tadi saya ceritakan itu juga on proses, sehingga nanti loginnya itu ataupun sistemnya itu mungkin tunggal ya. Jadi seakan-akan jadi satu sistem, nanti penggunaannya, kebijakannya nanti bisa shareable dan sebagainya. Kurang lebih itu ya infrastrukturnya. Jadi untuk eksternal ke dalam ada 100 giga, kemudian ada sumber daya komputasi macam-macam yang kita sediakan. Intinya ada yang multi nodes CPU, ada cluster GPU, ada storage gitu ya. Kemudian ada high memory node satu buah tadi. Jadi silakan kalau nanti ada kebutuhan dan memang cocok dengan kebutuhan infrastruktur komputasinya, kita akan berikan tanpa sistem ya. Jadi kalau kita mau buka, kita akan melalui ELSA. Jadi tidak begitu saja pakai gitu ya, tapi melalui prosedur gitu ya, supaya ada akuntabilitas dan juga ada log, ada pencatatan. Seperti tadi nanti bisa jadi pertimbangan manajemen BRIN, kalau penggunaannya penuh, ternyata komunitasnya berkembang, tentu saja nanti infrastrukturnya bisa ditambah. Nah, mungkin saya akan menjelaskan dahulu terkait penggunaan Mahameru ya. Dan ini sudah dipraktekkan sebenarnya untuk yang klaster yang ada di teratai. Jadi sifatnya infrastruktur terbuka untuk seluruh riset yang ada di Indonesia. Tidak hanya BRIN, bisa juga yang non-BRIN ya. Namun harus melalui ELSA BRIN untuk aplikasi akun dan juga aplikasi akses pada sumber daya komputasi yang khusus. Ya, jadi kalau misalnya untuk membuat akun itu silakan digunakan ELSA BRIN. Ini bisa saya tunjukkan. ELSA-nya ada di ELSA mana? Saya belum buka. Ada di website ELSA, kemudian sini ada BRIN HPC ya, di lain-lain. Agak ngumpet ya. Ya, nanti layanannya ini rencananya akan direstrukturisasi. Kalau saat ini hanya dibagi dua, layanan komputasi umum dan layanan komputasi AI. Tapi kita akan restrukturisasi berdasarkan jenis penggunaan ya, berdasarkan jenis penggunaan supaya nanti penyeliaannya itu bisa dibantu oleh teman-teman dari komite pengguna. Jadi nanti kita silakan untuk melihat akses layanan di sini, mengajukannya seperti ELSA pada umumnya, hanya bedanya di sini ada proposal. Proposalnya itu sebenarnya bukan proposal riset, bukan proposal yang kayak penuh gitu ya, tapi lebih ke apa sih pengen ngapain gitu, terus kita bisa lihat di sini ya. Jadi ada judulnya abstrak, tujuannya ngapain, perangkatnya apa, sumber dana apa, dan seterusnya. Jadi tidak ada file sebenarnya, isian saja, dan nanti penyelia akan menilai apakah permintaannya itu pas atau enggak gitu. Jadi kenapa kita butuh komite pengguna itu sebenarnya itu mengecek apakah permintaan dari user itu pas atau enggak. Jadi misalnya ada sebuah riset ingin menggunakan pendekatan data science misalnya, kemudian kasusnya adalah pengolahan data-data numerik yang table bentuknya ya, mungkin cuma 5 parameter. Kemudian dia mintanya GPU cluster. Tentu saja nanti bisa dinilai apakah ini berlebihan atau tidak. Jadi kita kalau misalnya, oh ini mah bisa di komputer workstation biasa, ya tidak perlu pakai akses ini gitu. Jadi nanti penilaiannya seperti itu. Tapi kalau misalnya ada yang mengajukan ingin mengolah data whole genome sequence dengan pendekatan data science, datanya jumlahnya sampai ribuan, terus satuan datanya bisa ukurannya besar, maka itu akan bisa dinilai ini layak untuk menggunakan GPU cluster seperti itu. Jadi penilaiannya tentu akan independen ya. Kami usahakan terbuka, misalnya kalau alasannya ditolak apa, kita akan ceritakan, kalau diterima juga gimana. Jadi pengelolaannya nanti berdasarkan ELSA, melalui ELSA, kemudian pengguna itu akan mendapatkan namanya ini partisi atau kalau bahasa queue atau antrian ya, atau partisi dari sumber daya komputasi yang sesuai. Jadi misalnya tadi saya ceritakan beberapa kasus akan butuh multi-node long time. Misalnya dia butuh beberapa node ya bersamaan karena paralel, dan selesainya bisa mingguan, harian, atau malah bulanan. Tentu saja yang kasus-kasus seperti ini kita akan lebih hati-hati memutuskan proposalnya ya. Apakah dia layak menggunakan ini atau tidak. Karena kan menggunakan resource yang banyak sekalian, dan akan ngeblok penggunaan yang lain. Jadi ini harus benar-benar diperhatikan pengajuannya. Tapi kalau misalnya hanya pakai satu node dan satu hari, tentu saja itu kita tidak lebih scrutinize, ya. Maksudnya defaultnya orang bisa akses itu. Nanti ya ini ada beberapa resource yang bisa dipilih oleh pengguna Mahameru. Kemudian terkait dengan softwarenya, tentu saja kita akan pasang, dan saat ini sudah terpasang beberapa software, termasuk yang untuk pipeline genomik ya, GHOST, nanti yang akan dipresentasikan oleh Pak Ajay. Modul software ini sifatnya dinamis ya, jadi bisa diinstal sambil berjalan kalau misalnya belum ada dan seterusnya. Akan disesuai dengan kebutuhan. Modul software ini bisa juga bentuknya menggunakan kontainer dan lain sebagainya. Nanti kalau teknis mungkin kita akan adakan workshop-workshop yang teknis, bagaimana nanti meload, memilih, menjalankan, dan seterusnya. Nah, modul software ini akan beda-beda untuk tiap jenis riset, mungkin akan beda ya. Pasti beda. Misalnya kalau orang yang biasa dengan climate, dia butuh software stack yang akan berbeda dengan orang yang bioinformatika, molecular modeling, akan berbeda juga yang AI, akan berbeda juga dengan yang material science. Tentu saja ini kita butuh masukan nanti dari komite pengguna, apa saja yang kita pasang dan seterusnya. Nah, kalau struktur pengelolaannya seperti ini, jadi lintas unit ya kalau di BRIN. Saat ini kita sebut manager lab ya, kemudian ada admin system dan admin software. Manager labnya saat ini adalah Pak Syam Budi Irianto, periset di Komputasi. Kemudian admin system, jadi yang mengelola user, mengelola storage, mengelola job, itu adalah periset-periset yang ada di OREI dan juga ada dari Ornamat ya. Dan kita juga melibatkan beberapa admin software saat ini yang berasal dari beberapa organisasi riset. Jadi ini pekerjaan community, dan kami butuh bantuan juga dari teman-teman periset yang memang punya pengalaman di HPC. Nanti silahkan kalau misalnya belum tergabung untuk bisa diusulkan. Kemudian kalau untuk infrastruktur jaringan dan pendukung data center ya, bukan data center dalam hal yang seperti di kementerian lain gitu ya. Ini kita minta bantuan dari teman-teman Pusdatin BRIN. Dan alhamdulillah saat ini sudah, misalnya infrastruktur jaringan mereka sudah bantu untuk membuat, kemudian menganalisis kondisi data centernya, itu sudah dilakukan. Tapi sekali lagi ini sebenarnya bukan data center biasa, lebih tepat disebut infrastruktur riset untuk komputasi. Karena kita punya beberapa alat infrastruktur riset yang bisa dipakai secara bersama-sama bergantian untuk kegunaan riset. Sedangkan kalau untuk infrastruktur HPC-nya sendiri, dari komputernya, PAC-nya, UPS, dan pengadaan dan pemeliharaan, dan juga kemitraan. Jadi kalau nanti di sini ada yang dari swasta ingin menggunakan, ataupun dari private sector ya, ingin menggunakan, ingin melakukan kerjasama, itu bisa melalui Deputi IRI, Infrastruktur Riset, dan Inovasi BRIN. Jadi kita ini memang beberapa unit yang bekerja sama untuk mewujudkan Mahameru, layanan Mahameru BRIN. Nah, dalam waktu dekat kita ingin juga membuat komite pengguna HPC BRIN, di mana komite ini tadi bisa menjadi penyelia, kemudian dia bisa juga yang membuat program, karena kan infrastruktur riset ini seperti grant ya. Jadi misalnya ada klaster DGX1, ada klaster tadi high memory. Nah ini atau multi node long time. Ini kan kalau kita berikan seperti grant sebenarnya secara in kind penggunaan infrastruktur. Nanti komite pengguna ini akan membuat SOP bagaimana pengaksesan infrastruktur ini. Umumnya sih seperti yang saya presentasikan tadi melalui ELSA dan juga ada verifikasi dari verifikasi dari penyelia. Nah, rencana kami itu akan terdiri dari beberapa unsur karena beda-beda tipe ya, tipe permintaan, tipe kebiasaan penggunaan komputasi. Jadi ada unsur ilmu hayati yang bioinformatics ya, baik itu analisis data omics ataupun molecular modeling. Kemudian dari unsur ilmu material atau fisika, misalnya yang biasa menggunakan material science modeling, misalnya quantum espresso, DFT, dan lain sebagainya. Kemudian nanti juga ada dari unsur ilmu kebumian, baik itu studi atmosfer, oceanografi, earth science, dan sebagainya, atau digital twin ya, kondisi real dari digital twin dari earth. Kemudian ada juga unsur yang dari kecerdasan artifisial dan big data. Nah, nanti komite ini secara umum kemarin sih arahannya bisa termasuk dari non-BRIN. Jadi kalau misalnya ada yang bisa mengusulkan dari universitas supaya termasuk member komite pengguna HPC BRIN itu dipersilakan untuk mengusulkan. Nanti SK-nya diterbitkan oleh Deputi IRI. Jadi komite pengguna ini nanti tugasnya merekomendasikan kebutuhan komputasi, memverifikasi proposal, terutama penggunaan sumber daya komputasi yang khusus ya, yang besar. Jadi ini kami mengundang siapa yang berminat untuk gabung supaya nanti bisa menjadi membantu dalam penyeliaan ajuan penggunaan HPC. Nah, kurang lebih sih seperti ini. Saat ini yang sudah ada, manajer lab sudah ada, admin sistem sudah ada, admin aplikasi sudah ada, tapi komite pengguna belum resmi dibentuk ya. Rencananya beberapa admin aplikasi bisa jadi komite pengguna juga. Nah, secara umum sih desainnya, terutama yang baru ya nanti kan ada, dan ini sudah dilakukan admin sistem, itu kita akan membuat beberapa partisi yang disebut publik, kemudian partisi yang terbatas ya, restricted, jadi hanya grup user tertentu yang bisa. Tentu saja nanti ini sifatnya diskrutinisasi oleh tadi komite pengguna. Kemudian ada partisi admin ya, ini partisinya nggak banyak, tapi ini harus ada untuk testing, untuk compiling, dan sebagainya. Tentu saja kita harus implementasikan software stacks atau modul. Nah ini sambil berjalan, ongoing terus, dan akan berbeda satu sama lainnya ya. Jadi kita mohon nanti kalau ada, oh ternyata kalau bioinformatik yang melakukan modeling kita butuh ABCD, ya itu yang harus disiapkan. Keempat itu nanti sambil berjalan juga kita melakukan integrasi ya tadi, karena sampai saat ini masih dalam proses pemindahan secara fisik, sedangkan secara sistem sambil berjalan. Dan yang kelima itu ini masih dalam kemungkinan apakah nanti kita lihat yang server-server dari Pekayon dan dari Junjunan itu apakah bisa untuk pengembangan lain ya. Jadi bukan sistem HPC tapi lebih ke misalnya untuk aplikasi, untuk server testnya ya. Tapi ini tentu saja kalau bisa jadi computing node lebih baik jadi computing node. Karena kalau ini sebenarnya di workstation biasa itu harusnya bisa. Nah, ini juga program kerja yang lain yang akan dilaksanakan terkait dengan diseminasi dan community building. Karena saya yakin kita perlu menumbuhkan komunitas pengguna HPC secara umum, sehingga banyak periset Indonesia yang punya kemampuan teknis ya. Tentu saja kami juga mencoba agar interaksi antara HPC dan pengguna itu semakin semakin tidak rumit gitu. Misalnya kita coba menggunakan paket-paket yang berbasis web untuk akses HPC. Saat ini mungkin nanti itu bisa menggunakan Plico yang produknya Lenovo, tapi itu tidak menutup kemungkinan yang lain ya. Jadi ada juga misalnya kita menggunakan PBS Pro ataupun yang lainnya. Nah, ini masih dipertimbangkan oleh admin sistem supaya interface penggunaannya itu tidak sekedar yang command line, yang mungkin buat teman-teman yang latar belakangnya bukan computer science akan jadi sesuatu yang sulit gitu ya. Nah, tentu saja kita dua arah tadi menyediakan akses yang lebih mudah, tapi juga kita ingin pengguna makin expert. Karena itu salah satu yang kami rombak nanti juga terkait dengan website HPC BRIN.id. Dan saya harap nanti yang komunitas itu bisa membuat panduan terkait yang spesifik gitu. Jadi nanti tidak perlu tidak perlu orang nanya lagi dari awal gimana makainya, gimana parameternya dan sebagainya. Jadi orang bisa membuka halaman panduan, kemudian bisa melakukan seperti itu. Nah, kami sudah organisasikan websitenya, tapi tadi pagi saya cek antara down atau tidak berjalan. Saya lagi menanyakan ke Pusdatin. Nanti ini juga akan dikelola secara berjamaah ya, maksudnya isinya gitu. Karena kalau semua dibebankan ke admin sistem itu tidak mungkin. Mereka juga periset, waktunya terbatas untuk menyediakan community service. Jadi yang bisa dilakukan ya kita melakukan bareng-bareng. Selain itu direncanakan juga workshop series yang sifatnya lebih teknikal. Nanti kalau sudah terpublish di ELSA, kita akan rencanakan workshop untuk pemula. Jadi barangkali kita akan kenalkan misalnya submit job dengan yang command line atau yang GUI seperti apa, kemudian yang teknis-teknis ya, pengenalan yang ya setidaknya bisa pakai gitu. Tapi bisa juga nanti ada workshop-workshop yang tematik sesuai dengan aplikasi. Misalnya nanti kalau ada contohnya yang biomolekular modeling, pengenalan GROMACS, flow untuk molecular modeling, ada GROMACS, ada yang buat ngitung energi dan lain sebagainya. Itu kita bisa workshop tematik nanti akan disusun oleh tim ya perencanaannya. Tapi yang salah satu yang mau kami adakan itu HPC School 2023. Ini saya belum hapus yang Oktober. HPC School direncanakan tuh minggu kedua Desember. Ini dalam proses ya. Mudah-mudahan lancar dan didukung oleh pendanaan dari Uni Eropa dan juga dari Fugaku Jepang. Nanti tracknya itu ada Life Science, Earth Science, dan Parallel Programming. Yang Life Science itu rencananya fokusnya ke biologi molekular modeling, kemudian Earth Science itu terkait dengan model untuk natural disaster, jadi dari modelnya sampai agent computing. Kemudian Parallel Programming itu lebih sifatnya coding ya, multinode, GPU, dan seterusnya. Nah, ini masih disusun programnya, open call-nya direncanakan pada akhir Juli, jadi mohon nanti silakan untuk mendaftar. Dan mudah-mudahan lancar ya, dan open call-nya tinggal dibuka dan teman-teman di sini bisa mendaftar. Nah, HPC School ini direncanakan dilakukan di Cibinong, di lokasi yang sama dengan HPC, yaitu Lab Indra Jauh. Jadi kurang lebih ini masih rencana dan kita akan publish kegiatannya melalui situs HPC dan juga lewat channel-channel yang lain. Ya, jadi kurang lebih seperti itu. Nanti tadi ini udah saya ceritakan ya. Jadi saat ini kita perlu memutuskan desain partisi dan storage karena setiap kasus beda kebutuhan dan kita perlu setting secara tepat. Kemudian software modul dan stacks ini sebagian sudah diinstalkan, tapi ini akan ongoing ya, continuous development. Dan dalam waktu dekat kita akan membentuk komite pengguna. Secara umum seperti yang tadi saya ceritakan, kalau user biasa ya bisa akses partisi yang umum minimal, tapi yang partisi besar, nah ini kita akan menggunakan komite pengguna. Dan penggunaannya itu selalu ada akuntannya ya, jadi terhitung misalnya menggunakan core hour-nya berapa, supaya kita bisa ada catatan, jadi akuntansinya akan selalu jalan. Namun saat ini belum ditetapkan apakah itu akan dihitung sebagai deduksi kepada ELSA point. Namun kita tetap hitung gitu ya. Saat ini masih dalam proses kebijakannya masih sifatnya berdasarkan nanti komite pengguna, kemudian kita akan cek penggunaannya, outputnya apa dan seterusnya. Karena kami masih butuh model perhitungannya supaya adil gitu. Karena kan nanti kalau misalnya kita tetapkan sama, kasihan nanti yang butuh computing node-nya lama, tagihannya bisa gede. Apakah mungkin dia baru bisa buat satu paper dari computing yang lama, tapi ada juga yang cuma sebentar tapi banyak yang bisa ditulis gitu. Jadi ini masih dirumuskan. Saat ini kebijakannya tidak mendeduksi ELSA point. Jadi follow up-nya ini mungkin mudah-mudahan ini lancar. Kita usahakan 17 sampai 28 Juli ini kita melakukan restrukturisasi layanan HPC di ELSA. Jadi kita akan bagi berdasarkan domain. Kemudian kita terapkan di ELSA itu penyelia-penyelia dan juga pelaksana, sehingga nanti bisa dibuka dan teman-teman bisa melakukan aplikasi penggunaan setelah ELSA-nya dibuka ya. Kalau saat ini kita masih dalam restrukturisasi, kurang lebih nanti memang kita menggunakan sistem ELSA supaya tadi akuntabel dan juga tercatat. Jadi kurang lebih itu pengenalan HPC BRIN saat ini, statusnya. Mungkin ada yang bertanya-tanya kenapa dipindah-pindahin, tadi saya sudah jelaskan alasannya. Nah, kalau ada yang ingin tetap bertanya, ingin diskusi, atau merekomendasikan komite pengguna, itu silakan ke email ke saya sendiri, misalnya Rifki Sadikin BRIN.id, atau lewat WhatsApp ya. Ini nomornya saya bagikan. Bisa juga lewat email HPC BRIN.id. Jadi itu saja dari saya. Terima kasih banyak. Saya kembalikan ke moderator Pak Adit.
Ya, terima kasih Pak Rifki.
warahmatullahi wabarakatuh.
Ya, terima kasih Pak Rifki atas paparannya. Bagi Bapak Ibu yang ingin menggunakan HPC Mahameru, silakan mendaftar melalui ELSA, dan apabila Bapak Ibu bersedia menjadi penyelia, silakan menghubungi Pak Rifki atau melalui email HPC BRIN yang telah diberikan tadi. Baik, kita mulai saja sesi tanya jawab. Mungkin Bapak Ibu ada yang ingin bertanya mengenai penggunaan HPC
Ini
BRIN.
dulu aja, Dit. Yang
Oh,
dari
ya. Dari
yang
itu aja.
dari nanti tanya jawabnya di belakang aja biar enak.
Baik. Kalau gitu, kita akan mulai sesi kedua, yaitu paparan dari Dr. Ajay. Mungkin saya switch ke bahasa Inggris ya. Okay, let us welcome our second presenter for today, Dr. Ajay Kumar Mahato. Let me share screen about his profile. Dr. Ajay, are you here?
Yeah, I'm there.
Okay, thank you, Dr. Ajay. Our second presenter is Dr. Ajay Kumar Mahato. Let me know if I say incorrectly of your name. He is a group leader of the Laboratory of Genomic Informatics from the Center for DNA Fingerprinting and Diagnostic, or CDFD, from Hyderabad, India. He joined CDFD in 2021, and his group will focus on research on bioinformatics, genomics, big data analysis, genomic resource and web application development, deep learning, machine learning, and artificial intelligence in genomics. And he previously led the bioinformatics team involved in India's first endogenously decoded plant genome, the pigeon pea genome, and other several genome like jute, mango, coconut, and others. And today he will give a talk about HPC for genomic peptide analysis. And maybe after a talk, we will have open Q&A. So please prepare your question during the talk, and then maybe use the raise hand function after the talk. Or maybe write your question in the chat. I will compile it and then ask Dr. Ajay or Dr. Rifki, and they will answer your question. Okay, now, please, Dr. Ajay, you can start your presentation. I will stop share my screen.
Thank you so much, Aditya and Dr. Rifki, for inviting me and having me on this online colloquium, which mainly targeted on the HPC and its usage, especially in the era of genomics. So let me share my screen first. Fine. Now it is visible over there.
Yes, it's visible.
Okay, fine. Let me just give me a second. So yeah. So for so good morning, everyone, and so it's 8:30 a.m. here in India, and I'm still in my home, so I'm attending this colloquium. So thank you so much for inviting me, Dr. Rifki and Dr. Yan. So the talk of my today's talk is title is HPC for high throughput genomics data pipeline and development and analysis. So especially it focused on the how you can use this high performance computing in the era of genomics. And myself, my name is Ajay Kumar Mahato. I am a group leader and a staff scientist at Laboratory of Genome Informatics. As Aditya, let me change the pointer to laser right. So this is the outline of my talk. So we are just going to discuss about basic things like what is computer, what is an high performance computing, what is the application, and the basics of genomics and bioinformatics. Why bioinformatics? Because once you have a genomic data, the next thing is the data analysis, where this bioinformatics come into the picture, is centered disciplinary fields. I'm going to talk a little bit about that. Then also from where this data is generated or generating, which require that much high potential capacity of computational requirement. And then followed by the limitations, future perspective, conclusion, and acknowledgement. So as you all know, and everybody is using computer, right? And especially smartphone is a kind of computer. which take an input, process, and give an output. So in a normal language, or in a layman language, a computer is basically an electronic device which stores the data, do the processing, and give you the output. The output could be desirable in your format in which you require, whether it's, let's say if you are searching on Google, so your input is text, but you're searching for image, so your output given by the Google search engine is a series of images. So I know how many of you know the full form of computer? So it's a basically common operating machine purpose, usually for technological, educational, and research purpose. So, and it consists of a CPU, which is the main brain of the computer, which contains the memory, RAM, SNPS, motherboard. And for the graphical user interface, you have a monitor for giving the input, and the we have two things, keyboard and mouse. So this is the basic what a computer it is. And what are the capabilities of a computer? So majority of things like me just, yeah. So the computer is capable of doing what? So first is operation. Everybody is doing operation, like calculation of data, manipulation, communication. The second thing is storing. The computer is widely used for the storing of information in the form of data. The data could be anything, whether it's a genomic data, your chat, your images, or your audio, your video. Data can be anything, which a digital form of information which can be stored is called a data. And execution. So we need for the execution, means for its processing, we need computers. And for smaller data set, the personal computer or the laptop you are using is enough. But when it's come into the bigger picture or a high throughput data set, this high performance computing is become a necessity, so that you process the data, draw the and you make the conclusion about that data set, so that you can further use for of your application. The application could be anything, like a research purpose, or like a training a model in machine learning, or anything else. And so the next come is like, what is HPC? So the HPC stands for high performance computing. So as you know, like if a single person can do a job in 10 days, then 10% together do the same job in a coordinated manner in 1 day, right? So so that is a basic concept behind the high performance computing. So it's a powerful system. So it's an interlinking of several systems into a single unit or entity to handle and to store, handle, and analyze large and complex amount of data. And it performance is also very important in that. So in this picture, you can see like this is an independent computer. This one is independent. But when these CPUs, they are stacked together and they are interlinked, it become a high performance computing kind of thing. So, and what are the characteristics of the HPC is basically it enables parallel processing, right? Where when you fire a job, it will the master node take the job, it will distribute the job to the job nodes or the slaves nodes, and the slave nodes do the calculation, and once the result is ready, it's sent back to the master node compile it and give it to you as an output. And by this way, the processing time is going to be very less, probably in a fraction. If it is earlier, if it is a day, it could be probably in a minute or in a fraction of seconds. So that is the power of computing, and especially high performance computing. And it utilizes the high speed interconnect. As Professor Rifki told, that this Mahameru BRIN HPC is a newly developed infrastructure, which is having the several servers with a high-speed interconnectivity, especially on the infinity band, where you can achieve 100 Gbps per second data exchange among the system. And it's also when it's compiled, so it's the performance is depend upon the memory. So any HPC on which you are working, if it is a 10 node, means 10 people memory, or if single node is having 1 TB, you have like 10 TB of memory in one go, in a one entity. So it gives you a large scale memory which can handle a massive amount of data, like big data, which are in GBs, TBs, or petabytes. So these are few you can say like capabilities of HPC, what it can do. It can process the complex calculations at a very high speed. It can deliver the exceptional computing power to process the data. And one of the best known type is like a supercomputer. So high performance computing is basically a supercomputer. So Mahameru BRIN HPC is probably is a supercomputer in which contain probably compute node which have like thousands of cores which are interconnected together, and which work as a single entity to complete one or more tasks. So this HPC, or you can say supercomputer, can able to run one task in one time, or it can be able to run parallel jobs in a one go. Okay. So if you see like, so the next question is, okay, if the high performance computing is so powerful, what are its main component? So the main component are the servers. So there is called, we will segregate server into two parts, like master node and compute node. The master node, its job is to only manage the compute node. Like whenever you fire a job or you submit a job on the master node, it will see its data throughput, and on the basis of algorithm, it will distribute the job to the compute node. And after once the compute node process finished and the result is ready with the master node, it will compile it and send it to you and give it to as an output. So these are the two important components. The third thing is like interconnectivity. So let's say in this example, this is a picture of an HPC facility. So in which you can see all greens, this light are servers. So these servers are they are connected in a stack. They are stacked, and behind that they are connected with the InfiniBand. InfiniBand give you the power of data exchange at a very high throughput scale, like 100 Gbps per second. Also, the storage file system is required. So the main you have to decide whether it's I hope this Mahameru is running on the parallel file system, or probably it's an object store system. The third important thing is like scheduling, like Slurm or job schedulers. Let's say in HPC, several people connected together. In this example, like 10 to 12 users are connected to a single HPC, and they're firing their job. Okay, so once, let's say if two person has fired their job and it is running, or all the resources are in the utilization or in the computing node. So the third person, when he or she fire a job, the scheduler read it first. Okay, whether is any compute node is free to run that application. If not, it waits until the resource is available. And once the resource is available, means other process got complete, it then send the job to the execution, right? And the third, like the important thing for the management point of view is like resource management software is required to monitor the health of your HPC, whether how much resource are used, whether the resource are used at optimal scale or not, whether the storage is sufficient or not, whether the temporary files. Sometime what happens in the home directory, if your temporary files got stored and it got full, your system got hanged and your system got crashed, right? So this is the major component, like master node, compute node, interconnects, its file system, and scheduler, followed by the resource management software. So in this just a picture, you can say a user is connected with the logging node, and he fires some job. It will go to the so this login node is a master node. So the master node read the job. Okay, it required, let's say 200 CPU cores and like some 6 TB of RAM. Then it distribute those jobs into several pieces and send it to the job nodes. The job node will process the data, and it will again send the result to login or master node. Master node reconcile the data and give it the output to the user. followed by it's a file system where the data is stored and executed. So there are huge areas of application. Now coming back to the application, that HPC is everywhere, whether it's a computing, whether it's a genomic, especially personalized area, computer designing, drug discovery, machine learning, that material science, remote observations, like and the IoTs. So everything is basically governed by the HPC, probably. Like if technical computing, we are talking about medical devices, and the IoTs are especially connected with the work on this aspect. So when you are talking about the interconnect or IoTs, so like I will give you an example of Alexa. So if you have Alexa or any other like Amazon Fire in your system, and if you are giving any command from your resource or some other area, so first it will go to its server, and then from the server the information goes to your device. It look like that you are directly connected to the device, but it is not. Behind that, which you cannot see, is the high performance computing, which manages lakhs or millions of such kind of IoTs over the internet. So this is the huge area. But coming back to the especially genomic research and the personalized medicine, yes, without HPC these days you cannot do the genomics research, right? I will give you one example of COVID. Every whole world is suffered with the COVID, and the vaccine development is only fastened due to this high computational power and the sequencing facility. Like any variant which is pathogenic or non-pathogenic, it is easily detected using the amalgamation of genomics, precision genomics, and the HPC. Because let's say if you have a tons of data, like terabytes of data, but you don't have that much kind of high performance computational power, so you cannot get analyzed. It takes time. Hence, the HPC is coming to the picture, and it is now becoming the core or heart of the of any application in this era, and it is going to be the future also. Because of the coming of AI, it is very important. So to go further, I'll just give you a brief overview, in past 2 years, this is my lab, which I have developed because I joined 2022. Before that, I worked for 13 years in a national Indian Agriculture Research Institute, which is the biggest institute, or it's a center in India which governs the agriculture research. So currently we have like 32 servers, which include one dedicated server for HPC, which called like 24 node HPC. We have a 6 node cloud-based server also, where we are offering services. We have a storage of 2.5 petaflop. This is the real picture. This is DDN high speed 2.5 petaflop of storage capacity, which include, and total thread of the CPU is itself like 1826 cores. Coming back to the NVIDIA GPU, as my lab is especially working on genomics and the bioinformatics, especially on the big data and AI. So we have procured the NVIDIA DGX A100 blocks with fully populated, what this Mahameru BRIN HPC is already having the same setup. But we have taken this pack, NVIDIA Clara Parabricks, which is specially designed to make use of GPUs power for doing the SNP calling on a large data set. As my research institute is basically, we are focusing on the human health sector and the fingerprinting, and my lab is running on the one of the biggest program on pediatric genetic disorder, where we have generated 6,000 pediatric rare genetic disease patient sample, and to call the variant on the human genome. You see, the human genome is 3.2 GB of long. To map those data, we require high computational power. So we have procured this NVIDIA DGX A100 fully populated to get analyze our data. And yeah, it's really fast. That is one job, and it has a multiple application. So as we have, my lab is also working on some AI algorithms, which can able to differentiate the male chicken with the female chicken, which is very important, which is a major problem in poultry in India, where it's very hard to identify in the initial state of the chick, whether it's a male or female. So we are working on that. We got a special grant on that. Also, this HPC is also working for the whether several genome project. So this is about the basic overview. We are just giving services also, like pipeline, as this talk is basically on genomic pipeline. So bioinformatics pipeline we are giving, and with a secure, we have a workshop. We are developing the databases. We are offering cloud services, and as well as we offer the consultancy also. So coming back to the main picture, so how HPC look like is this. So this is you call a supercomputer, okay, or a HPC. So there are so this is a representative image which I have taken from this paper. So let's say, and you are a user. So let's say if you are a biomedical researcher or a PI or a collaborator or a material science researcher. Okay, now you have a big data, or you have to run simulations like Professor Rifki told, like GROMACS and all those things. So GROMACS works very well in GPU. So you are not directly connected to the HPC, right? So you got the terminal access SSH. You are connected. User is connected via the SSH, and there he or she fire a job. The job is going to the managed by the master node, and it decides whether the data, the job should be run on the GPU mode, or GPU servers, or the CPU servers. And it in MPI mode, it distribute a job. And once the job is finished, it will recompile and send the data to the user. So in a fraction of minute, you got a, oh, I have like some 200 GB of data, and within 1 hour I will got my result. So that is the power of supercomputer. But if you use a normal workstation or any high-end workstation, it will end up with an error called like out of memory, or the core is insufficient. So that is one part. The second part is like the data storage is also very important. Everyone is talking about the processing, but what about the storage? The data with what we are generating is a very important. Anyone's like in here, over there also, like in Mahameru or in Indonesia, I'm going through papers now. You guys are working on the genomics especially. So the data, keeping the data is very important. So this can also act as a file storage, which can manage the control access. So this is the power of a supercomputer or high performance computing, which has two components. One is processing. It will offer a high speed processing capacity to the data set. Plus, it will manage the data, large volume of data in a secured manner, so that you can retrieve the data efficiently over the FTP secure environment or anything, or any media via SSH. So, yeah. So basic about like, as this lecture is like, genomics pipeline. So genomics and bioinformatics. So basically the genomics is nothing. It's an entire genome, study of entire genome, which include the identification of genes and its sequence. Okay, that is called genomics. And the genomics have a number of application or a number of field called omics. I will talk about omics, all the omics later in this lecture or in this talk, especially. And the bioinformatics, yeah. So the bioinformatics is the buzz of the research domain, and it is basically what? It is a computational tool to analyze and interpret genomic data. It's not a genomic data. It could be a proteomic data. It could be a mass spec data. It could be image data. But yes, without a good bioinformatician who is having the and better knowledge of computer science and statistics plus programming, it is not possible. And just mark my words, there is huge scarcity of trained bioinformatician pan globe, and this could be going to be a very good career option in a upcoming future in the era of AI, because in the era of AI, everyone is sitting on the big data sets, and having that much the fusion of the computational knowledge, the statistics, and the biology is going to play a very critical role. Especially the job opportunities in pharma sector is increasing day by day, and especially on the research domain also. So there are many tools which people are using, like for high throughput sequencing, for transcriptomics and proteomics. But this all genomics, it enables to identify genes that are functional or that are responsible to play an important role, whether the gene is causing a cancer, whether the gene is responsible for the improvement of a crop yield or crop nutrition. Okay, and this genomic is used for the development of molecular markers, not just used for the breeding program, but for the precision medicine, but for the human health, right? In our CDFD, so we have developed designed molecular markers which are able to identify several genetic linked disease. So so it helps us to do the prenatal testing, and if the baby is suffering or the fetus is having some genetic abnormalities, we will tell the parents, okay, look, this is the SNPs or markers which are coming positive. Probably your child is going to be it's not well, and it is having some deformality. So if it's better to do the abortion and all this. So by this way, we are saving the society. We are saving the pressure. We are just lowering down the pain of a parent who is suffering, whose son or whose child is suffering from any rare genetic disorder. Right. So genomics is basically what? It's a when we talk about a genome. So a genome is a complete set of gene which includes the genes, the non-coding region, and the mitochondria or chloroplast genome. So that is called genome. And the study of its sequence basically is called the genomics. Okay, there are several aspects like structural genomics, functional genomics, and the comparative genomics. This particular image, you can see here, the genomics start from here. It has several omics, like epigenome, where we are seeing the interaction. Transcriptome, where you are identify whether, like, what are the genes which are expressed in a normal condition and stressful condition. Okay, so that, and that proteomics of, let's say, if that person is having a normal and diseased person, so what are the protein level changes in their genome. So that comes under the genomics, right? Metabolomics. So what are the metabolite changes, microbiome? So all the omics today is that play a very critical role in the field of data analysis, especially in genomics. Okay, and this, this genomic term was coined first by Tom Broderick in way back in 1986, when the human genome is just now initiated in that era. Now, so that is all about the genomics. Now, how this data is generating. So this slide has two parts. One is like shotgun sequencing, another is next generation sequencing. So earlier in the 80s, when like human genome, so it was done using the shotgun sequencing, but it's very tough, it's very time consuming, and also it requires too many manpower, because it has a cloning step, it has genomic DNA extraction, everything, and the only one run you will generate only 66 or 96 reads or fragment of DNA sequences. But once after 2008-9, when the next generation come into the picture, there is no space for the this cloning. So cloning was removed, and you have to just take your biological sample, do the isolation fragmentation, and just give it to the machine, and machine will give you the data. But the data here is generating, it's in the GBs, right? And currently it is now generating in TBs. I'll give you the example. The latest machine called Illumina NovaSeq 6000 plus, in a single flow cell, it generates 4 terabyte of sequence information. Can you imagine? And if 2 flow cell is fully populated, it is generating 8 terabyte of the data. So you see the data. So to store the data, to analyze the data, you need a HPC. Okay, coming back to the lecture, this is the like a next generation sequencing machine which generate that huge amount of data. There are many of this, like this Roche is now obsolete. Iron Torrent is basically used for the targeted resequencing. ABI Solid is now obsoleted, but this is the future, packed by helicose and nanopore. So in our institute, we have Illumina NextSeq 6000, which generate the 4 TB of data in one run. And we have a nanopore also for the longer read. So the basic difference between these two machines is, one is short read, another one is a long read. So why? And after that, it has low in cost. The data generation time is very less. Now, this is a table which summarizes, right, the first generation, second generation, and the third generation of the sequencing. So you see in ABI Sanger only 900, only like you are able to generate like some some 96 reads. Now it coming back to the second generation, it become 1 million, 25 million, 3 billion, right? And for the longer read, for the shorter read, and for the longer read, it become increasing day by day. And the basic difference is between this technology, second generation and third generation, is only the read length. You can say the average read length is varies here. This is the important point. Like in the second generation, the 444 offer like some 450 base pair of read. But now in this PacBio, you will able to get some several KBs. Average now we are getting some 90 to 25 KB average KB length of the reads. And you know why I have written this? High computational power and trained human resource is required to analyze that kind of data, and the entire world is flooded with the data. But there is a huge demand of a trained power human resource who can able to make sense of that data. And the expertise needed is like he should have like the terminal Linux experience, Python programming, little of statistics, and shell programming. This is basically a graphical representation of what is bioinformatics. It's basically an field of science in which all the field like computer science and IT technology and biology is merged. Now, this slide is important, and please pay your kind attention. So bioinformatics can be used as a technology, and it can be used as a methodology. So means the data science, especially genomics, can be used as a technology, and it can be used as a methodology. So just coming back to the technology. So let's say when you have a let's say if you have DNA sequence, so what are the operation you can do? You can do the gene finding, which require high computation. You can do the comparative genomics. You can do the evolutionary analysis, right? You can do the and once you have RNA-seq or like the RNA data, you can do the annotation, differentiation, expression analysis. If you have a protein, right? So here we are talking about the technology. So by using bioinformatics technology, you can able to identify the structural prediction, do the docking. You can run the simulations. Okay, if you have a molecular network, so you can do the protein-protein interaction, which is basically a transcription regulation network metabolite. You can able to identify which are the networks which got upregulated or downregulated during the interaction of an infection. Okay, so and finally you can able to link it with a phenotype. So a gene can be identified, sequenced, annotated, and finally on the basis of this phenotype and genotype association called GWAS analysis, you can easily you can say, okay, this gene particular SNP is important for that phenotypic variation. So this is about the technology. Now in terms of methodology, like I told you now, the HPCs have two parts. One is analysis part, another one is storage and management part. So here, if you have a huge data, so the data management is a very critical. Like, and you know, data management is not simple like putting and dumping a data. The data should be managed in a structured manner. Like it should have a metadata information, a backup copy, everything. Okay. Once it comes to the you have a data, but in competition, you need so there is opportunity open for developing new software, new algorithm, new web servers. Like we have developed several web servers and application, especially who can do the SSR mining, who can predict the SNP and in the mitochondrial genome, which are responsible for any genetic diseases. If you have a, let's say, data mining, so people these days, NCBI, ENI, it's a flooded with the data. Okay, if you don't have if you don't want to generate data, there is huge data which is still remain in the public domain. You can make use of that data to draw new information about that particular problem, and you can publish it also. And yes, like predictive models and simulation modeling. So if you have protein data set and you want to see, like I will give you a drug discovery example. If any drug, if somebody is working on drug and for making the permutation, combination, and simulation, so that HPC play and methodology, this bioinformatics play a very important role. So this is how the bioinformatics is act as a methodology also. Now, I'm not going to talk much about this, as I have already spoken. That application area which require the HPC, like if anybody is working on genomics, so the broader objective is like he or she might be interested in decoding of genome or structural predictions or SNP SSR variation or expression pattern when if you are dealing with RNA-seq data, right? If anybody is dealing with the proteomics data, so structural modeling, structure designing, drug designing, especially an interaction, find out whether the protein-protein interaction or protein DNA interaction is happening or not. Like in case of gene chipset, like in system biology, when where your main focus is to do the pathway analysis, and in the phenotype is like image analysis. Now, due to this coming of this era of AI, so I'm not talking about the chat GPT, right? I'm talking about the artificial intelligence and several other models and machine learning. This image analysis is now improving day by day. And my lab is working on the image analysis for the characterization, for the differentiation of male and female chick from a group of chicks. So image analysis but it required a very high computational power, because you have to design a model first, and then you have to have a positive set and you have to have a negative set. Then you have to design the model, train the model, and so all those process is a very high computational and storage intensive. In this point of time, the HPC come into the picture. So this is a schematic diagram what I prepare for you. So let's say if you have a genomic data like genes and DNA, right? And using this software and HPC installed computational algorithm, what are the other things you can do? You can do this number of things. You can do the pan genome study. You can do comparative market discovery. Let's say if you have a transcriptomic data here, right? So using this data set, and with the help of HPC, you can decode a full transcriptome. You can do the differential gene expression analysis, which will help you to identify the responsible genes, and which will helpful for you to do the varietal improvement in terms of agriculture sector, right? And also you come to, in the human health sector, you come to know, okay, these genes are differentially expressed. So maybe these are the disease causing genes. If you have a protein like proteomics, so all are omics example. What are the application using? What are the data analysis or application you can do? Like you can do the complete set of protein expression by cell, or 3D structured modeling and drug designing, right? Let's say if you have a metabolomic data, you can identify the involvement of metabolites, which is basically produced during that chemical process and pathways. If you have a secondary data, now the secondary data is the very important thing, right? So so all these genomic, transcriptomic, proteomic and metabolomic data set, they are generating using sophisticated machines, like, okay, we generate data in petabytes and gigabytes. But the secondary data is in where my lab is working, okay, because we make sense of use this data. The data is present is where the data is present in the public domain, okay, but using this big data analysis, clustering management tools, or clustering algorithm, okay, machine learning and intelligence, we are able to draw new information with the same data set, because the secondary data set provide an opportunity without even spending a penny on data generation. Using the publicly available data, you can do your research, and yes, you can design the product also. I will show you one example which will help you to understand how. So this is the enlarged view of how my lab, or like you can use the secondary data to do this clustering, all those things, but it requires the high computational power. And mark my words, this Mahameru BRIN is going to be a game changer for the Indonesian research domain. As per what I have gone through, it's a specification, and especially it's a very good opportunity for the users and for the researchers to get trained on the sophisticated cutting and technology, and just trained and do good, not for the science but for the humanity, right? So Yeah, so this is a very important and a very interesting slide. So this is Moore's law of data growth, right? Moore's law is given by in 2000, early to 2000, which says whenever as the time progresses, the cost of data generation is going to be less, right? But the throughput is going to be increased. So here you can see the example. So before 2007, okay, when the people are working only on genomics, especially, or other the data generation like human genome or rice genome, mouse genome, the data is generating using the Sanger sequencing data set. So that time the cost is very high, but you know the data throughput is very less. Okay, but once this next generation come into the picture, like Illumina and all those things, you see the data is the throughput is increasing like petabytes, right? But you see the cost is going to be very less, from $10,000 to it become $0.1. So this is the power of Moore's law, and this trend is going to be followed for next 10 years, where people are sitting on a data, but the main thing it requires is the computational power and the trained human researchers who can able to handle that data and make use of that. Even though this open API or like Chat GPT, it is not going to change this domain, because it requires a scientific mind who can think, who can assess the output which is given by a machine, right? It cannot automate the data processing, because I tell you one thing, because if you are running let's say one genome assembly on one tiger genome, so the parameter to assemble the tiger genome not going to work when you are passing a rice genome with the same software on the same parameter. You have to have tuned the parameters, right? Which AI cannot do, and where this human resource, trained human resource is required. So even though the technology has its this Chat GPT or like Open API has cut down several jobs, but yes, if you are a good computational biologist, or you know how to work on HPC environment, your future is secure. So this is the current trend. So why I kept this slide, these two slides in my presentation, because this I just want to make aware of the students also that, okay, what is the current trend in globally? So the globally everyone is working on genomics, like genomes, functional genomics, metagenomics, single cell genomics, artificial intelligence, genome editing, right? So here you see the data generation is very, very easy, right? But the bottleneck is the data analysis, right? So the data analysis where, because there's a huge scarcity of trained human resource who can able to handle large scale of data, or even who are able to work on the terminal, right? So after this, if you go and search just postdoc position or PhD position in bioinformatics or in computational biology, you are flooded with the pan globe, and there's huge demand, but the trained manpower is very less. Hence, there is a scarcity, and the pay is also very good. So, what is the requirement for that one? You should have a proficient in Linux environment working in command terminal, SSH or VPN. You should have a good programming in Shell. Shell is basically a program. So my lab is mainly using Shell and Python to make the pipeline. Pipeline is nothing. It's basically you are linking several steps into one script and fire it. So one by one, the software itself, after completion of one step, the output of that step is become input of secondary step, and second step start working. So this is the basic of a pipeline. So if you have this kind of expertise, your future is secure. Yeah. So this is an example I'm giving. So like it's about a genome assembly, right? There is a world biggest project is going on, whose name is like Earth Biogenome Sequencing Project. And mark my words, the objective is to the sequence all the living species which is present over the earth. And there are many countries, like some 50 countries already the partner of that consortium, but they don't have that. But now they require the manpower who can do the data analysis, and that point of time they are struggling. Even US there is huge demand, UK there is huge demand, in Europe there is huge demand of trained manpower who can able to work on this environment, etc. So nothing is what Sanga, if any data which comes, it will go to the software, right? And the software, if it is a HPC, you have to run on a job scheduler. The scheduler decide where on which node it has to send the job. Once the job is completed, it will give you the data set. So the assembly is basically work is the backbone is like the sequence similarity, like 1, 2, 3. So this fragment of DNA is equal, but this fragment of DNA has a little bit of that another fragment. So by this way, on the basis of consensus, a small piece like 1 kb, when it's going to join, it form a complete entire genome. So I'm not going to talk more about this sequencing, but it's another domain. But I'm just showing you the power and the requirement of HPC, where it is required. So this is an algorithm view. You see, so there are fragment, like billions of fragment of a genome. Let's say if you do a Illumina sequencing of a genome, let's say who's having 430 MB at 100x, it means you are generating 443 GB of data, which is coming in pieces. So these data set need to be fed in a software, and it cannot run on the normal workstation or high-end workstation. It required HPC. So in HPC environment, as it is having a huge RAM power as well as the storage, it will able to handle that one, as because it works on MPI mode also, and it will stitch all the data and give you the genome for after the analysis. And also for the variant calling, this mapping is important. If you have a large, like thousand of genotype or patient sample, it cannot be done on the your normal computer or HPC. So for that, like the NVIDIA DGX A100 box is a very good option for you, especially Dr. Rifki, to make use of that to do the high-end mapping. So it saves your time, it resource, and so pretty it's a pretty fast. So these are like, as I work mainly earlier on the breeding part. So this is one slide which I have kept for agriculture student. So what are the applications assisted breeding you can do? You can identify the region in the genome. You can identify the casual genes. You can identify the germplasm, which influence the phenotype. The sequence of genome and transcriptome enables. Let's say if you are decoding a genome, like I got an opportunity and I'm fortunate to work for my country, and I'm a leader in 5 and 6 genome sequencing projects, and I represented international also, where the main goal is to identify the genome plus identification of marker, which can be given to the breeder for faster breeding, and so that the crop variety can improve. So, and the earlier it is even taking 9 years to get a new variety. Now within 3 years you will have the new variety, but it requires genomics and the high computational requirements. So this slide is also what you can do: gene editing, genomic selection. I'm not going to much more, but yeah, trait mapping identification is basically a development of a molecular marker. When I'm talking about marker assisted breeding, means a precise, efficient, cost effective time reducing developing of a new variety. Already everything is governed by the genomic or omic data. For genomic selection, like you are identify some trait which controlled by the multiple genes. When gene editing, so to identify the SNPs, like CRISPR-Cas, using this technology, you can do the editing. And yeah, genome association study, you identify the haplotype of a gene which is responsible for that phenotypic changes. So now future perspective of genomic breeding, yeah, it's basically going to be omic based only, but it has still a very vast area of developing new algorithm and software, where this HPC play a very important role. And also in this era of AI, it's a very one of my collaborator, we are working together to just correlate the genotypic, the SNP variation which we got from the genomic data with the images of the crop infection. So probably it could be a new kind of dimension we can give, where we are using the AI and HPC with the genomic and the image analysis to do the early prediction, so that we can save that one. So this is the basically some examples I just kept, like how this genomic can change the entire scenario. So this rice genome is very important. Like this QTL identification, it may arise by variety tolerant to heat, right? And this particular gene identification is production of high yield. So it means like this genomics is having the power to not to address only the human health or precision medicine, but yes, the food is the main important. Okay, if you are hungry, you cannot do the science, but with a full stomach, we can think. So there the genomics play very important role. This is the gold mine. If like anyone who is working on rice genome, this rice genome project is a gold mine for the rice researchers. You see, this rice genome was sequenced like in way 2002, right? And it has gold standard. Takuji Sasaki from China, so he is from Japan, he lead it. But still, the like many, like 70% of the people still working on the rice genome. So you see what is the importance of genome. But yes, now they are making use of this high performance computing. So that is the gold mine. Similarly for the maize. Yeah. Now coming back to the limitation and challenges for the genomics is the big data management and analysis. So guys, the important thing is that the challenges is that where people are not aware, they don't know how to manage, how to deal with a large amount of genomic data set, how to use the high performance computing power for doing their analysis. Okay, they rely on vendors, give the data to vendors. Vendors will give anything, and okay, you believe. But the problem comes when you are validating in your wet lab. Whatever marker they are developing, it is not working. Or even when you are communicating a research paper, if some reviewer asks for this analysis to be redone, you don't have any idea, because you have to depend on the third party or vendors. So by this way, Professor Rifki, especially I'm just trying to say you that this is a very important era, and this is a very important domain where the students, scientists can develop their expertise, right? And by doing so, even though they are not dependent on the vendors more, but they can do the cost cutting, which they can further use for other research purposes, right? And yes, it requires expensive equipment and expertise. So you guys, you are fortunate, like this Mahameru BRIN HPC is the lifesaver for you, where you can do n number of things. Material science, if you have you have to run the molecular dynamics simulation, you have a GROMAX, you have GPU cluster, you have a CPU cluster, you have a large storage, you have Ghost-based. Ghost-based is also, Lenovo Ghost-based is also a very important and a very good servers, or you can say technology, which is developed by in collaboration of Intel and this Lenovo, which can offer you two things. One, if you want to do the snip calling, it will give you GPU like performance in a CPU manner. Plus, once you are not using that one, you can use that server. I hope this Ghost base and Ghost Plus, the minimum server store RAM is like 1 TB or 2 TB. So 2 TB server, 2 TB RAM machine is very good for doing the genome coding kind of stuff, or running the stacks pipeline. So by this way, you are in a very good infrastructure, and I just request you make use of it, learn it. Right now, the second thing is ethical issues is important. So this ethical issue is like the safety. You have to use very HPC should be used in controlled manner. The administrator should be vigilant, because these days the cyber attack is happening. Okay, and the majority of cases has happened, and this it is happening during the insider threat. Insider threat is nothing. So they're the insider people who knowingly or unknowingly they click the link, and it will spread in your network and infect your HPC also. And once your HPC, if let's say ransomware attack got attacked, you have to rely on that. So sensibly use that one. Use the proper protocol, whatever SOPs is there to use the Mahameru. Now, coming back to my last point, like what is a computational pipeline? So till now I have talked about like what is genomics, what is bioinformatics, what is an HPC, what is a computer. So to summarize till now, a computer is normal electronic device which can store and analyze the data. HPC is basically a series of computers joined together, which work in an interconnected manner in a single entity to perform high throughput data analysis in a less span of time and do the management and processing. While the genomics is basically this era is a genomics, as the sequencing is cheaper, is cheaper and cheaper. The precision level, ATGC level research is now booming. It is not the genomics, it's a metabolomics, proteomics, secondary, and you can say like this microbiomics is also coming up. So those are technology for the data generation. Now, once you have a data, now the next step is what you have to make sense of that data. So for that, you need a high performance computing facility or HPC. So what Mahameru is going to provide you, you make use of it, and then use the HPC in a very controlled and efficient manner. Plus, follow the S&P. Now what is a pipeline? So let's say a pipeline is basically a series of computational steps that perform a substitution analysis and generate an output. I will give you a live example in my coming slides how I have developed a pipeline where I have used like public domain data, and I have developed one physical high throughput genotyping array, which is now under the process of patent. So basically, it's a series, it's a computational step where you are interlinking at a logical level, okay, and where you are just breaking a complex problem, bigger problem into smaller problem, like some random forest approach you are using. And this is used to streamline and then automated complex data analysis process and processing tasks. So once a pipeline is ready, you just fire the pipeline. It will automatically take the data, do the analysis, do the computation, do the filtration, and produce you the result. Plus, it send you the SMS or email. Okay, look, your job is finished. Please download your result from this link. So this is how a pipeline works. It composed of multiple interconnected modules, like each responsible for specific task. So, okay, and the pipeline often involve data information, filtration, analysis, and visualization. So now, if the pipeline, so what is the main component of a pipeline? So, Dr. Rifki, is I'm running out of time or what? Just give me 10 minutes.
No problem. You can continue your presentation.
Thank you so much. So the component of computational pipeline is what? The first is data. If you have a data, then only you are able to analyze. So the data is important. So it is obvious a raw data, files or streams. It could be anything, right? It's a binary data, it's a image data, it's a sequence information data, it's a phenotypic data. Data could be anything, image also, voice also, videos also. Now, the next is data processing. So once the data you have a data, you have to pre-process the data, which require like the cleaning, filtering, transforming into a suitable format so that the software can able to accept it. So I will give you one example, the sequencing data which is coming from the machine. It is in a FASTQ file format, right? And the data which is coming and is a raw data, it contains several cells. It contains several adapters and the poor quality basis. So for that, the pre-processing is required. Okay, so the pre-processing, so and every step is depend on the software. So pre-processing done by a software like Trimomatic or FastX. Okay, similarly, so the second step is data processing. Third is the major step is data analysis, where you are using several softwares or the designated software to do the analysis. So I will give you one example. Let's say if I'm my job objective is to do the genome assembly. So I'm going to use a software called SOAP-DNO. So the FASTQ data first it will do the undergo in the processing, pre-processing, removal of poor quality and adaptive sequence, and after that the filter data is input in the data analysis software to extract the meaningful result or to do the assembly. Okay, but during that analysis, when algorithm is running, okay, it create an intermediate files. So which is a very large size. I say if you are inputting a 2 GB of data, the intermediate files could be goes up to like some 100 fold also. But it depends the algorithm. But on an average, if you are giving 1 GB of data as an input, the intermediate files which is generated, probably it is 15 to 20 GB. So you can imagine that much data, it has to be stored intermediate, and that is in the RAM also, and in the temporary folder. So this intermediate data storage is also required. So you have to define where this intermediate data is going to be stored. After that analysis, the data visualization. If something is there, we have to visualize. So the step is visualization, then output generation. Okay, everything is fine. Now, these are the FASTA files which represent the genome. Okay. So this is the component of a computational pipeline. Now, as I told you, so this is an example which and this paper was published in Nature Scientific Report, where we have developed the 62K SNP chip. And so earlier in the previous slide, this is on the visual part or verbal part. Now this is a graphical part. So this is a live example where we have taken 17,000 some odd genes, and we use the RAD sequencing and 46 variety Illumina pool sequencing, okay, for mapping. So the first one is this aligning and mapping. Then it's conversion to SAM to BAM format. Then pile up SNP calling. Then filtration, and further it will go with the filtered SNP, which we have sent to Informatics USA, and the chip was designed. And because this work has taken my so I have invested 9 months to automate this one. It's not a single straightforward job. You have to sit and you do the pipeline. You have to, every step you have to do the running and checking, and everything should be run properly. So now this pipeline is standardized, right? So using this pipeline, we have developed 80K chip for the mango genomes, and some 67K chip for the amaranthus genome. So it has, even though it has taken my 9 months to get the pipeline done, now my previous institute, so they are using it. So so they are analyzing like some 700 GBs of data. In 15 days, they are ready with a format which they can able to send it to Informatics USA for the fabrication. So this is how a pipeline make your life easy. You don't have to sit in front of computer, okay? Okay, after this step, I have to fire this job. A pipeline is contain everything in one file, text file, and it will sequentially do the operation, and it's based on the logic. So that physical chip we have crossed. So this is the chip which we designed for the Pigeon P, Professor Rifki, and we have validated. And the data which is coming, it's a super. You can see it's a clear cluster of like the haplotype A, B, and AB. So this is how you can make use of the data set. But behind that, why we are able to do that is first two things. First is because of that high performance computing what we are having in our previous institute, and yes, the pipeline which automated all the function using that. Now now it become our daily routine. You can develop the chip like things from that script, and there are a number of applications for that chip. Okay, so it has using that data set, you can do the linkage mapping, you can generate the linkage map, you can do the GWAS analysis, you can do the haplotype genome gene analysis, and many more. So to conclude, so next generation sequencing is so advanced, right? So it gave you the flexibility to generate terabytes of data without cloning and PCR amplification. So everyone is now increasing the data size day by day, but for the analysis or making sense of that data, HPC is required. It's a must-have thing, whether it should be central or it should be institutional. And the best thing, beauty of this HPC is what? So let's say if this Mahameru BRIN HPC, it is located in one location, and via the SSH or like a VPN access anywhere entire world, if I'm sitting in India, I want to make use of that your HPC. I can connect it via the secured VPN, via SSH tunnel, and get my job done over there without coming down there, right? So by this way it is going to be a central hub where because it's management and its management cost is very high. So if it is a centralized place, so whole Indonesian or any research institute who don't have that capacity, they make use of this. So this is important thing. Now, and genomics and big data, sorry, it's a bog data, it's a typo mistake. It took a trained human resource. Hence, yeah, why I'm just making emphasize a good career option, because every day, like I'm getting every week some mails from my collaborator who are in UK and Europe. Ajay, do we have any good bioinformatician who can able to work on the HPC environment, and they can do this some SNP calling, because they are working there in a precision medicine where the major objective is to do this SNP calling. And like you guys are fortunate that in Mahameru you have this Lenovo that server, okay, which is able to do this SNP calling, okay. Yeah, it's a data is a gold mine, but it requires understanding and ban from this approach. And the future genomes look promising, right? And but the only thing is required is the expertise, understanding, and the ethics to work on HPC, because I have seen several labs here who they don't follow the ethics or SOPs. Due to that, there's a lots of data breach, and this data thing that got corrupted. So this is basically going to be a responsibility of a HPC administrator who is going to handle the Mahameru BRIN HPC. But yeah, in totality, the coming era is for the genomics with the amalgamation of high performance computing. And this trend is not going to be stopped. Like coming 10 years is very much secure, and it's bring more job opportunities for the bioinformatician or computational biologist. So with this, I'm very much thankful to my director in charge, Dr. Sangeeta Mukhopadhyay, Dr. Jan Reinto, Director of Laboratory Genome Management Research Facility and Science Technology BRIN, and yeah, of course, Dr. Rifki, for providing me this opportunity to give a talk into such a nice audience. Thank you so much, and I'm open for the questions.
All right, thank you for Dr. Ajay for his true and insightful lecture. So next, we start the Q&A session. You can use the raise hand function on Zoom, and then I will give you the space, and you can ask any of our presenter any question. You can say your name and the affiliation, and then who do you want to ask, whether it's Dr. Rifki or Dr. Ajay, and they will answer your question. Please, you can raise your hand now.
So Arif Priyadi.
Okay.
Okay, thank you for the opportunity. I'd like to ask to Pak Mahato. Is it okay?
Yeah, please go ahead.
Okay, it's very interesting to follow your presentation, and I'd like to ask if you don't mind. I mean, how many libraries? I mean, you showed in your presentation that we have many different machines like Illumina, Oxford Nanopore, and so on. So far in my work, I mean, I just use one platform, the Illumina, the Illumina short read. And after we try to assemble de novo assembly, this is from the genome of plant. We extract the DNA from leaf, then we try to de novo assembly. The de novo assemble the reads, but we've got a short context, and when we try to search for the literature, you cannot just use one library and you need another one. For example, you need to sequence with Oxford Nanopore to make a better scaffold and so on. And can you please explain more on this subject? Thank you very much.
Thank you so much, Arif, for asking this important question, right? So this problem we faced with early in 2009 when I started working on the Illumina data sets. So the problem with the Illumina data set is because it's a short read, right? And it cannot able to address the homopolymeric region in the genome. So what happened? You will end up with a short means large number of contigs. Okay, so the thing is that for your solution, if you generate some nanopore dataset, Oxford nanopore dataset, or like in this current era, like Packboy Hi-Fi, which will give you the Q40 type of read, where the average read length is probably like some 18 KB to 20 KB. So you can imagine in Illumina 100 base pair you are assembling. Now you have an 18 KB, which will be going to a backbone, and you can use Illumina for polishing of that data. Or you can use the latest technology called nanopore, make a backbone, do the polishing with Illumina, you are good with the genome. Okay, so I hope your objective is to decode that genome. Arif, is I'm right?
Yes, yeah, and can I mean some paper says that we can use.
Yeah, Arif,
reference.
one more thing. Let's say if I will give you one example, right? So I'm interested to sequence a rice genome, right? Of my example. Let's say I'm taking a black rice of my area. I generated the data similar to your only Illumina data set, right? And I assemble the data. Now the number of contig is very high. It is in thousand, some 10,000, 20,000, 40,000. It doesn't give you any clue whether which contig is represented by which chromosome, right?
Yeah.
But the alternative is that, Arif, if you can map those contig on the rice genome reference genome,
okay,
and
yeah.
do the scaffolding, right? And this scaffolding is going to be unbiased, and at the end of the analysis, you are left with the like 11 chromosome of rice and few contigs which are unassembled. So by this way, your unordered piece of contigs, it arrange in a chromosomal fashion, right? Why? Because both are rice, right? So rice is rice. Okay, 99% of genome are similar.
Only thank you.
So by this way, you can make use of it.
Okay, thank you.
Thank you so much,
Pak Aditya.
Okay, thank you, Arif. So next question. If you have trouble in speaking English, you can also write your question in the chat, and so I can translate it to Dr. Ajay. Okay, Bu Maulida, please.
Okay, thank you. Can you hear my voice?
Yes.
Is it clear? Okay, thank you. Yeah, I would like to ask to Dr. Ajay. It's very nice presentation. Yeah. So my question is, do you have any experience working on system biology using HPC? And if you have, can you tell to us? And for example, for pathway analysis and network biology analysis, what tools or what method that we can implement using HPC? Thank you.
Okay. Thank you so much, Maulida, for asking this question. So when you're talking about the system biology, so basically network is going to play an important role. You have to find out like, so we are using cytoscape here, basically, for identify the network, and also string. So these two software we are using, and we are using it for in one of the crop called pigeon pea, which is a legume, and but yeah, it is not it is a GUI mode. So you have to work on your not on HPC. But system biology, when you're talking about the large data set, when you are going to run it, okay, whether it's ortho finder or anything else, that can be you run on HPC in MPI mode. And one more thing, like I'll just convey to everyone, so you don't think that every software will going to work on the MPI mode or like message passing interface, because HPC backbone is message passing interface. If a software which is compiled, which is written in a message passing interface, then only it make maximum use of HPC. Or else, if the software is not written in MPI mode, it will run in a single node. Okay.
Thank you for your clear answer, Dr. Ajay. I think it's clear now. Thank you.
Thank you, Maulida. Thank you so much.
Okay, any more question? You can also ask Pak Rifki for anything about Mahameru. You can ask him, and he can answer you in Bahasa Indonesia. You can ask him in Bahasa Indonesia. Okay, please, Pak Syam, you can ask your question.
Okay, thank you. I just want to ask to Dr. Ajay. Thank you for a nice presentation. My question is there are two questions. This first one is, I was wondering what job schedule did you use? And I mean, is it like SLORM or PPS or etc.? And is it using terminal based or there is a graphical user interface? The second one is regarding to the pipeline. For instance, for certain analysis, I mean, what pipeline framework did you use? Usually used in your group? Is it like SnakeMake or Nextflow or etc.? Thank you.
Okay, thank you so much, Nyanto. So we use slums in our HPC to fire the jobs, and basically all the students, they are using connected via the SSH link or the terminal mode. And for the pipeline development, as you know, like this Slurm and SnakeMake. So earlier we are using SnakeMake. We are using now for making our bioinformatics computation pipeline.
Okay, thank you.
Thank you.
Okay, next question. Okay, maybe because there's no more question, maybe I will ask Dr. Ajay the final question. Because I'm from the background from the file programming, can you tell me about where in your tools or bioinformatics tool that you use in HPC that have bottleneck? Maybe the bottleneck is in the storage, or maybe in the memory usage, or maybe in the computation itself. So can you explain to us where's the right now in your experience, where's the bottleneck in the current bioinformatics tools?
Yeah, Aditya,
Yeah.
very nice question. Very nice question, right? So yeah. So as during my talk, I discussed like so my lab is working on the decoding of a chicken genome, and that genome is 1.3 GB now. We have generated some 200x of the data, which is amalgamation of the PacBio Hi-Fi data, Illumina data. The data is probably about some 900 GB of data. So when we use Pylon, which was not configured in MPI, okay, so because our one node is having 384 GB of RAM, single node is having 384 GB of RAM, and we have 24 node HPC. So whenever my students fire a job, so it runs for few days, but it got stuck. So then when we try to find out what is the problem, so then after 1 week he identified the bottleneck is that whenever the intermediate files where this OLC, because it's a Pylon, it's work on the OLC, overlapping consensus approach, whereas the data is of long read. So it don't have that much memory to keep the OLC the intermediate data in it. So it cannot able to hold, and that's why it give an error. So that after this problem, we suffer for 2 months, then we procure, then I just ask that, okay, we have a Ghost Plus which is having a 2 TB of RAM, and so why not you are using it? So the beauty of Lenovo Ghost Base or Ghost Plus is what, whenever you want to use it for the GATK pipeline, you can use it. Otherwise, no, rest you can make use as a single node server, which is having an 2 TB of RAM, and we install the Pylon and we run, and it got completed successfully in some 6 days. And but on what, yeah, so that is one success we got. But yes, as I told you, on one parameter you cannot get the optimal assembly. So we ran some 3 tools, Kanu, TrioKanu, Fly, and from there we chosen the best one on the basis of the BUSCO analysis, telomere to telomere repeat identification. So the bottleneck comes on the data set and the type of application you are. If your data require, or your application is single machine, and it cannot be pass their data information in MPI, you cannot make use of HPC. So for those complicated programs or data execution, you need a FAT node. So this one bottleneck we got. Now the second bottleneck, which I hope like it was already addressed in Mahameru, is the data backup. So Professor Rifki, because earlier, so we have a 2.5 petabyte of primary storage, which give throughput of 40 Gbps, and it has an infinity band connectivity. But we don't have the DR side, let's say, because we lost some 20 GB of data. So then we just now established 5 petabyte of secondary data storage, that just for the data archival. So why? Because Professor Rifki and Aditya, so whenever you run a high computational jobs, it runs for a month, right, or few weeks, and during few weeks it creates some intermediary files, right, and to generate the final output. So let's say if your output is okay, but when the reviewer asks you, you just rerun that analysis, okay. But if you deleted the intermediate data, you have to just invest that many more days. But if you have that data ready with you, you can directly change the parameter and run it. So, and yes, that is one thing. Second thing is data repository and DR site is important, because students can do they do the error, and we cannot stop them. Sometimes they will remove the rm-rf command, which will remove everything. So in that case, we should have a backup of that thing. Okay. So these two bottleneck I face, and we are in the process of fixing it.
Okay, thank you very much. If there's all the question, let's give a clap with direction for Dr. Ajay. Okay, thank you very much, Dr. Ajay. And maybe Dr. Rifki have some closing statement.
Okay, thank you, Adit. I will deliver the closing statement in English and Bahasa. In English, actually, I just want to say thank you for your presentation, Dr. Ajay, Mahato. It's a great pleasure to having you here. I hope we can still continue to have thing like research collaboration in the futures. Dan buat Bapak Ibu sekalian hadirin, saya perlu mengingatkan bahwa HPC Mahameru BRIN ini dirancang untuk generic application, jadi tidak khusus untuk bio. Jadi kita infrastrukturnya bisa mendukung bermacam-macam kebutuhan komputasi ya. Jadi dari material science, life science, AI, dan juga climate and earth sciences. Kebetulan hari ini topiknya ya memang genomik, jadi bukan berarti yang lainnya tidak difasilitasi gitu ya. Jadi terkait dengan pembukaan dari HPC Mahameru yang baru, kami akan lakukan dalam 2 minggu ini pembentukan juga komite pengguna tadi. Jadi kalau ada teman-teman yang punya pengalaman sebagai HPC users yang expert dalam hal yang mengetahui ya bagaimana computing resource itu dibutuhkan untuk specific case, bisa gabung dengan komite tersebut yang nanti bisa merumuskan tadi model komputasi ataupun storage sistemnya itu seperti apa, dan juga bisa nanti kita mengadakan semacam bagaimana metode untuk verifikasi ya penggunaan dari pengguna. Jadi kami terbuka kalau ada input dan saran, silakan bisa hubungi langsung ke saya ataupun lewat email. Saya juga mengucapkan terima kasih untuk moderator Aditya Swardiana dan Bu Yetty selaku MC. Terima kasih ke Bapak Ibu semua. Saya kembalikan ke MC untuk menutup acara ini secara resmi. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Baik, terima kasih Pak Wayan Aditya Swardiana M.Kom yang telah memandu sesi pemaparan dan diskusi yang sangat menarik. And thank you to Dr. Rifki Sadikin M.Kom and Dr. Ajay Kumar Mahato for the great presentation. Baik Bapak Ibu yang berbahagia, alhamdulillah kita telah sampai di penghujung acara. Terima kasih kepada seluruh peserta kolokium yang telah menyimak dari awal hingga akhir acara. Kami mengingatkan kembali kepada Bapak Ibu untuk mengisi presensi yang sudah di-share oleh panitia di kolom chat. Dan untuk materi hari ini akan di-share oleh panitia melalui email peserta. Saya Yetty Lestriani, mewakili panitia yang bertugas menyampaikan mohon maaf apabila ada kekurangan atau kesalahan selama acara berlangsung. Izinkan saya menutup acara dengan sebuah pantun. Jalan-jalan ke Kota Batu, bersama teman dari Bengkulu. Sampai jumpa di lain waktu, semoga sehat dan bahagia selalu. Terima kasih Bapak Ibu, thank you.
Terima kasih Mbak
Terima
Yetty.
kasih Pak Rifki.
Thank you Dr. Ajay.
Pak
Thank you
Aditya.
Ajay.
Thank you.
Terima kasih Bu Lili ya.